So thank you, everyone who is not running out for lunch, and hopefully I'm going to keep you interested in the next, in this talk. So thank you for the organizers for inviting me to share today. And so I'm really excited to tell you about some of the work my lab's been doing in trying to understand sex-dimorphic regulation of innate immunity with aging. And so thankfully, all the previous speakers in this session have explained a lot of concepts. I'm not going to have to go in detail in a lot of those, but I want to sort of hammer in some even deeper, something that Sarah just highlighted, that not only people don't necessarily annotate sex in their data, but still in the aging field, when we go and we reanalyze the data, and you can actually tell transcriptomically if a sample is female or male. So even if people don't tell us, and we get the data, over 3/4 of the data sets we've looked at end up being strictly from males. And that's a problem because, again, at the end of this session, I'm sure you're now convinced that females and males age differently, but because we don't have basically the molecular data, we don't understand really the pathways that can regulate those differences. That's a problem because longevity interventions have responses that are extremely sex dimorphic. Like we know that things like 17-alpha estradiol only work in males, and there are things that work much better in females than in males. What that tells me is that if aging was the exact same thing in both sexes, then you would expect that the same intervention should have the same effect, but it doesn't. And so that means there are some fundamental differences that we need to work on and understand. We also know, and again, thanks to Dr. Voskel for highlighting that, age-related diseases skyrocket in prevalence after menopause in women, showing again that those kind of things need to be included. Thanks to the NIH roadmap of genomics data, there's now been shown that even in tissues people don't necessarily think of as sex dimorphic, like the liver, the biggest source of differences in terms of gene expression and epigenomics is the original biological sex of the donor, not the age, not the BMI, not the disease status. And then finally, and that's something that Dvigu Ushar showed today, is there are huge differences in the immune responses and the susceptibilities of males and females. And so that's why we got really interested in this. And my lab does work both on neutrophils and macrophages, but today I'm going to tell you about what we're doing with neutrophils. I want to make sure to give a shout out to the people doing the work in the lab. Ryan Liu, grad student, Minhu Kim, postdoc, Cassie McGill, who is showing another project today. And I think it's also important to always sort of highlight the people who gave you the idea in the first place. And so I want to give a shout out to Shalina Taylor, who told me neutrophils were interesting and that they were, that they needed more attention, and without whom I wouldn't even have looked there. And so, I'm a neutrophil convert, and so I want to convert all of you as well. Neutrophils are this really weird, but really cool cell type. They're also the most abundant white blood cell that you have. 50 to 70% of leukocytes in healthy humans are neutrophils. And so you would be like, okay, so everyone must be working on them. There's so many of them, right? Right? No. The problem is they're a pain to work with, because they're extremely short-lived. On average, once a neutrophil finishes differentiation, it has less than 12 hours to live. The longest lived neutrophils live 4 to 5 days, but they're very rare and they need to go in tissues. And the big joke in immunology is like, you look at them sideways and they activate. And so it's really a problem to work with, so you need to be really careful when you work with them. They're very characteristic, so this is just H&E on a blood smear, and they have this really weird nucleus shape. That's why people call them polymorphonuclear cells, but it's actually a single nucleus. It's just packed in a really weird manner. And it's probably linked to the very interesting biology of neutrophils, where they've evolved the capacity to extrude their chromatin at extremely high velocity, forming those fibers that you can see, or maybe not. Well, on the electron microscopy, those fibers are actually chromatin fibers that can trap microorganisms. And so those neutrophil extracellular traps are necessary for life. Like if you're not able to have neutrophils that do their function, that's something that David Gems was highlighting a few sessions ago. If you don't have neutrophils, you can't survive. But if your neutrophils are overactive, it actually creates a lot of problems because then you're going to have circulating DNA. So, everyone knows about the anti-DNA antibodies that are a problem in autoimmune diseases, but also this can create a state of very exaggerated inflammation that is very deleterious. But so again, those cells are a pain to work with. So not many people have, tried to look at them with aging and with sex. And so we were really interested in figuring out whether they were affected by organismal aging at all, and whether this might be regulated in a sex-dimorphic fashion. And so we had access to the NIH mouse colony. And so we got young animals, and we chose 4 months because that's about where estrous cycle is very stable. And so that's basically, not looking at a teenager, but an actual adult. And then old mice. We then, because that's the most abundant source of neutrophils in mice, extracted the bone marrow and then purified neutrophils to perform an unbiased multi-omic assessment of their phenotypes at the transcriptomic, metabolomic, and lipidomic layers. And so this was one of the first big omic profiling experiments across sex and age we did in the lab. And so I didn't know quite what to expect at the time. And in hindsight, I shouldn't have been surprised. But at the time, we were really surprised by how big the sex differences were. And so in this case, basically what we're doing, anytime you have high multi-omic data set, it's very high dimensional. Human brains can comprehend that. So, you project them on a 2D space, and that allows you to see, what are the big axes of differences. And so we're doing this here using a technique called multidimensional scaling. And you can see in this case, it is very clear that we can separate the female and the male samples, respectively in pink and blue, very easily, no problem. And then maybe if you wink a little and check in the diagonal, you can separate the lighter colored, which are the young animals, to the darker colored dots, which are the older animals. So, there might be a secondary here age effect. If we look at the metabolome, then there's a completely different picture where we can still separate the females and the males very easily, but the axes for aging in the males and females are actually almost orthogonal. And then for the lipidomics, we sort of have a intermediate thing happening where we can still separate females and males very well. There might be, if you squint a little bit, there might be an aging separation, but it's not very good. And so consequently, when we perform the statistics, we can get detect genes that are significant as a function of age and sex, but we can only detect metabolites and lipids changing as a function of biological sex. And so this is an aging conference. So I'm gonna, focus on the aging aspect, but if you're interested in the metabolomics and the lipidomics, it is published, and our analysis of this is published. And so every time you see big differences in the starting space of it, in the young animals, there is a base difference in the female and the males. One of the big questions we always ask in my lab is, okay, well, is there also a difference in trajectory? Or maybe they start from a different space, but they have the same trajectory with age. And so the naive way we look at this is by comparing here on the x-axis, the log-fold change in gene expression with age in the female neutrophils. And on the y-axis, we're looking at the log-fold change in gene expression for all the genes in the transcriptome in the male neutrophils. And the idea is that if those trajectories are very similar, the majority of the genes should be on that red diagonal, and the correlation should be very significant. And so the correlation is actually so significant, the computer can't even calculate a p-value, so that's why it returns 0. So we have extremely high correlation here, suggesting that the trajectories of transcriptional aging are very similar here between the female and the male neutrophils. I want to here put an aside, I'm not showing you the data for time, but we're looking at other cell types like macrophages, and that is not the case in macrophages. The trajectories are very different. And so I just want to highlight, this is probably going to be very cell type specific. Now, if we look at the same data, but under a different angle, which is, okay, well, what are the genes that pass significance in one sex, but not the other with age? You can see the large majority of them are male genes in this case. And it's not that they're completely uncorrelated. It's like the slope is higher, right? And so what we think is happening, at least transcriptionally, is that those male neutrophils might have an accelerated transcriptional aging phenotype. Now, the fact that we have a very good correlation is great because it gives us additional statistical power in the analysis of our transcriptome, because it means we can look at the effect of age while regressing sex and the effect of sex while regressing age. And obviously, I'm a biologist, so all the omics is great, but what I want is the function. What are the different functions? And so if we looked at this, 6 minutes? Okay. Looking at the big pathways that are regulated with age, we have pathways up and down with age. I'm going to concentrate here on the ones that are down, where there's an overwhelming representation of cell cycle and chromatin related pathways. And this was really weird. The reviewers asked us to double check our neutrophils weren't proliferating. So I can tell you for sure they are not proliferating, but we think that might be linked to a different way of handling their chromatin. Similarly, when we were looking at what was going on with biological sex, the females and the males, the majority of the pathways and actually the majority of the signal was coming from things that were more male biased. And if you look at the names of those pathways, you will notice that the majority of those pathways are also chromatin and cell cycle related. And so at this point, we were thinking, okay, well, maybe there's something funky about the way chromatin is organized in old neutrophils or in male neutrophils compared to the other groups. And so to answer this question, we decided to take advantage of a technique called ATOC-seq that allows the profiling of both local chromatin accessibility, but also gives you a view of how chromatin is organized, because it can detect nucleosome-associated chromatin, nucleosome-free chromatin, and so on and so forth. So for the interest of time, I'm not going to explain exactly, but this is one data point that represents all the data we have on analyzing the ATOC-seq, where we're looking at the occupancy of nucleosomes at expressed genes. And so here we're looking in the young and the old separately, but you can see that in both cases, the pink curve is significantly under the blue curve, which is consistent with the notion that there is an increased nucleosome occupancy in the males, regardless of age. Now, when we look in each sex separately with the effect of aging, we're noticing something kind of weird if you've been listening to the heterochromatin, loss of aging hypothesis, which is that the darker curves are significantly above the lighter curve. And that suggests that the nucleosome occupancy and thus the chromatin compaction is increasing with age, regardless of sex. And that is weird because it goes against everything we are told about how biology works, but that's sort of a leitmotif with neutrophils. They never work the way you think they're going to work. So again, for the interest of time, I'm not going to go over that. But the idea is that we had those differences in chromatin regulation. It looked we had differences in terms of the chromatin organization. And so since we thought this might be linked to their ability to do netosis, so to extrude their chromatin, we decided to look at that. And I think that's sort of a cautionary tale in terms of, predicting how things are going to be interacting functionally when you're looking at big data, right? Like here we were saying, with aging, the same thing is happening in both sexes, separately. With sex, the aging effect is the same, so they should interact in a predictable manner, right? No, they don't. What we noticed when we tested the ability of neutrophils to undergo netosis is that if you just look at the females, there is an increased capacity to do netosis. So it means they're more activatable, which is not going to be great because it's going to increase the inflammatory responses, a lot and create potential tissue damage. But if you look at what's going on in the males, yeah, maybe if you look just at the median, you can be like, yeah, it's kind of going up. But then you look at the variability of it, and there's not really an effect of age in the netosis capacity in the males. And actually, it goes to the point where the young females have the lowest netosis capacity of all the groups, including the young males. But that is completely flipped in the old, where the old females actually have the highest netosis capacity. So we were really interested that there was definitely an effect on mitosis that we had predicted, but we didn't predict that weird interaction there. And so neutrophil biology is very sex-dimorphic. That goes all the way to the chromatin architecture, which is a fundamental biological property of those cells. If you're interested about all the details of that first part, this is published, and we're now trying to understand hormonal versus genetic drivers of those differences. I want to give a shout out to the lab members because they're the ones who do the work. Fantastic collaborators or sources of funding. You for your attention, and sorry for going over time.